Every year, small birds embark on journeys that span continents and oceans, arriving at destinations they have never seen with unerring precision. The European robin, along with species like silveryes and garden warblers, performs this feat using a navigation system that has fascinated scientists for decades. These birds sense the Earth's magnetic field and use it as a guide. But what makes their compass truly remarkable is how unlike ours it is. The avian magnetic compass does not point north and south the way a handheld compass does. Instead, it detects the inclination of magnetic field lines, the angle at which those invisible lines dip into the Earth. This tells a bird whether it is flying toward the equator or away from it, essentially showing it where it stands on the globe. Even more intriguing is where this sense lives. The hardware is not buried deep in the brain or distributed throughout the body, it sits in the bird's eye, meaning these animals may literally see magnetic information overlaid on their visual world. But this compass has strict requirements. It only works under blue light in laboratory experiments. Birds placed under red light lose their ability to orient entirely. The system is also surprisingly fragile. In another way, it operates within a narrow band of magnetic field strengths, centered on the natural geomagnetic field. If the field becomes too weak or too strong, the compass falters. Unless the bird has time to adjust, this magnetic sense is not unique to birds. Salmon navigate vast ocean distances to return to their birth streams. Sea turtles cross entire oceans to find the beaches where they hatched. Honeybees and lobsters and fruit flies all carry some version of this hidden perception. Yet, the avian compass stands apart in its sophistication and in the strange constraints under which it operates. Those constraints, particularly the need for light and the peculiar way it reads field lines, have led researchers toward an explanation that defies ordinary biology and ventures into quantum physics. For decades, scientists have debated how birds might sense magnetic fields. One leading theory pointed to magnetite, tiny magnetic particles embedded somewhere in the bird's body. But another possibility emerged, one that places the magnetic compass squarely in the eye and draws on the strange rules of quantum mechanics. The radical pair mechanism begins with a simple chemical event. When light strikes certain molecules in the retina, it can trigger a reaction that tears electrons apart. Normally, electrons exist in pairs spinning in opposite directions, but when this reaction occurs, each molecule ends up holding onto a single unpaired electron. These two molecules, each carrying its lonely electron, form what scientists call a radical pair. What happens next defies everyday intuition. Those two unpaired electrons can become quantum entangled with each other, existing in what physicists call singlet or triplet spin states. In a singlet state, the electrons spin in opposite directions. In a triplet state, they spin in parallel. And this is where the Earth's magnetic field enters the story. An external magnetic field can influence how these electron spins relate to each other. It nudges them ever so subtly, causing them to oscillate between singlet and triplet states. This oscillation determines which chemical products ultimately form. The outcome depends on the angle between the magnetic field lines and the molecules involved. In classical physics, magnetic fields are far too weak to influence chemical reactions. The energies involved are minuscule compared to the thermal energy that typically drives molecular processes. But quantum mechanics operates by different rules. The radical pair mechanism exploits quantum coherence to amplify a tiny magnetic signal into a meaningful chemical difference. It is a process that should not work according to classical chemistry, and yet it may be happening inside the eyes of migrating birds right now. Now we zoom in on the actual molecular machinery that makes this quantum compass possible. The star of the show is a protein called Cryptochrome-Based. Specifically, the version found in European robins, scientists refer to it as ERCRI-4a, and it sits at the center of this entire navigational system. What makes this protein special is a molecule embedded within it called a flavin chromophore. This flavin is not permanently attached. It is held in place through what chemists call non-covalent interactions, meaning it sits snugly in a pocket of the protein without being chemically bonded to it. This arrangement turns out to be crucial for what happens next when blue light strikes the flavin chromophore. Something remarkable occurs. The light energy kicks an electron loose, and this electron transfer creates what scientists call a radical pair. Remember, those entangled electrons we discussed. This is where they come from. The flavin and its partner molecule each end up with an unpaired electron, and these electrons remain quantum mechanically linked to each other. The Cryptochrome-4 protein is not floating randomly through the bird's body, it is concentrated in the retina of the eye. This location is deliberate and elegant. By sitting in the retina, the protein can directly influence the bird's visual system. The leading hypothesis is that the magnetic field alters the chemical products of these radical pair reactions, and these changes somehow create visible patterns that the bird can see. Imagine looking at the sky and seeing a subtle overlay that shifts as you turn your head, always aligned with the Earth's magnetic field lines. This is why the compass only works in blue light. No blue light means no radical pairs form, and the entire quantum sensing mechanism goes silent. The protein essentially acts as a transducer, converting invisible magnetic information into something the visual system can process. It is biological hardware designed to bridge quantum physics and perception. When blue light strikes the flavin molecule inside cryptochrome, something remarkable begins to unfold. The flavin becomes photoexcited, creating an electron deficiency that sets off a precisely orchestrated chain reaction. But how does an electron actually reach this excited flavin? The answer lies in a molecular relay race. 4 tryptophan amino acids line up in a row within the protein structure, forming what scientists call a tryptophan chain. These residues act as stepping stones for electrons. When the flavin needs an electron, it pulls one from the nearest tryptophan, which then pulls one from the next, and so on down the line. It is a sequential handoff, like a bucket brigade passing water toward a fire. Researchers used a technique called ultrafast transient absorption spectroscopy to watch this process in real time. What they found was astonishing. The electron transfer does not happen all at once. Instead, it unfolds in 3 distinct stages, each with its own characteristic timing. The fastest component occurs in just half a picosecond. The second takes about 30 picoseconds. The slowest stretches out to roughly 150 picoseconds. A picosecond, by the way, is 1/trillionth of a second. Each of these time constants corresponds to one of the 3 tryptophans positioned closest to the flavin. The nearest one hands off its electron almost instantly. The next one takes a bit longer. The third requires even more time. The 4th tryptophan, sitting farthest away, appears to play a different role entirely. But how did scientists prove which tryptophan was responsible for which timing? They used a clever approach called phenylalanine substitution. By genetically modifying the protein to replace specific tryptophans with phenylalanine, a similar amino acid that cannot donate electrons as easily, they could knock out individual stepping stones one at a time. When they removed the first tryptophan, the half picosecond signal vanished, remove the second, and the 30 picosecond component disappeared. The experiments confirmed that each residue plays a specific predictable role in this electron cascade. Evolution has tuned the distances and orientations of these residues to optimize the transfer process, creating a biological wire capable of moving electrons with extraordinary efficiency. One of the most elegant ways to test whether quantum mechanics really powers the avian compass involves something surprisingly simple. Scientists can swap out atoms, not just any atoms, but specific isotopes within the cryptochrome protein. This approach works because different isotopes have different nuclear spins, and those spins directly influence the hyperfine interactions that shape how radical pairs respond to magnetic fields. Think of it like replacing strings on a musical instrument. Change the tension or mass of a string, and the sound changes in predictable ways. Similarly, when researchers computationally modeled what happens when hydrogen atoms in the flavin chromophore are replaced with deuterium, a heavier isotope of hydrogen, the results were striking. The magnetic sensitivity actually improved. The anisotropy, which is the directional dependence that makes a compass useful, became stronger and showed altered symmetry. This uniform deuteration effect makes a clear prediction. A bird with deuterated cryptochrome should have an even sharper magnetic sense, but the computational work went further. The researchers also explored what happens when carbon atoms are swapped for carbon-13 at specific positions within the flavin molecule, at 3 key locations known as C4, C4a, and C8 alpha. This substitution had the opposite effect. It dampened the anisotropy, making the magnetic field effect more uniform and less directionally informative. These predictions matter because they give experimentalists a concrete roadmap. If you want to prove the radical pair mechanism is real, you now have specific targets. Deuterate the flavin and watch the compass sharpen. Substitute carbon-13 at those positions and watch it blur. Either result would constitute powerful evidence that quantum spin dynamics, not classical chemistry, drives this remarkable sensory ability. So we have these electron spins dancing between singlet and triplet states inside the cryptochrome protein. But here is where the story takes a remarkable turn. The fate of those spins determines what kind of chemical product emerges from the reaction. When the radical pair recombines in a singlet state, it produces one type of molecule. When it recombines in a triplet state, it produces something different. And here is the crucial insight. The Earth's magnetic field, by influencing those quantum spin dynamics, effectively tips the balance between these two outcomes. Now imagine thousands upon thousands of cryptochrome molecules distributed across the bird's retina. Each one is running its own radical pair reaction, and each one is subtly influenced by the orientation of the magnetic field lines passing through it. The result is a shifting patchwork of reaction products across the back of the eye. The leading hypothesis for how this becomes useful information is genuinely startling. Scientists believe these reaction products may directly modulate the sensitivity of the light receptors themselves. Areas with more of one product type would respond differently to light than areas with more of the other. The bird would not perceive the magnetic field as some separate sense. Instead, the magnetic information would be painted directly onto its visual field as patterns of brightness and darkness. Think about what this means when the robin turns its head. The angle between its eye and the Earth's magnetic field changes. Those patterns of light and shadow would shift accordingly. The invisible field lines that wrap around our planet would become visible to the bird. as a dynamic overlay superimposed on the ordinary world. It sees. It would be like having a heads-up display built into your eyes at the molecular level, one that has been refined by millions of years of evolution.